Last time we were talking about the relationship between an amino acid side chain and how that side chain impacts the conformations that that amino acid will adopt as expressed in the phi and psi angles that we learned about last time. And one of the things I illustrated for you is that changes in side chain structure alter conformation. For example, alanine, which just has a methyl group as the beta carbon, is pretty comfortable in an alpha helix, less comfortable in a beta sheet conformation. Somebody say wait, am I supposed to wait? Okay, all right, good. Hold on, slow down, right. The only reason we know this is alpha helical and beta sheet is because we've looked at the structures, measured the dihedral angles, and dihedral angles in this area are associated with beta strands and beta sheets, whereas dihedral angles in this area are associated with alpha helices. In any case, if you add methyl groups on the beta carbon, you have branching on the beta carbon that increases the amount of beta sheet conformation that that amino acid can adopt. So that's an example of side chain structure influencing conformation and function. I think I pointed out you see a similar thing if you look at the amino acid serine, pretty comfortable in an alpha helix, a little bit more comfortable than alanine in a beta sheet. But if you now add a branching methyl group on the beta carbon, you get the amino acid threonine, and it is much more comfortable than serine in a beta sheet. So you could infer from this a somewhat general principle that amino acids that have branching at the beta carbons are frequently found in beta sheets, and that's true. There's one more amino acid that has branching at the beta carbon, and that's isoleucine. And you can see that, like valine and threonine, it's pretty comfortable in a beta sheet. I think we also said, you'll notice that most of these amino acids have phi values that are negative, and most of them don't explore the positive region of the Ramachandran plot, except for glycine, which is weird. Glycine lacks a side chain, and so the lack of that side chain opens up this whole right-hand side of the Ramachandran plot. Glycine can now adopt a whole bunch of dihedral angles that the other amino acids can't, and that's because it simply doesn't have a side chain here that causes steric clashes. So glycine's more flexible, and it's comfortable adopting dihedral angles here and on the other side where none of the other amino acids are very comfortable. Okay, one other weird one is proline. Proline is weird because in the side chain, the alpha carbon is bonded to the beta carbon. That's not weird. But that beta carbon is tied back to the backbone amide nitrogen in a 5-membered ring. This is the only amino acid where the alpha carbon and the amide nitrogen are connected via the side chain. And it's this 5-membered ring that does that. Compare the dihedral angles for proline for the dihedral angles for alanine, and you can see that alanine adopts a wider range of phi values than proline does. Alanine's phi values can range from around -60 to almost -180. In contrast, proline's dihedral angles for phi are all focused around -60. It can't get to -180. That's because of the ring. The phi angle refers to rotation around the bond between amide proton and alpha carbon. And you could build a model and show that the ring prevents you from rotating around to dihedral angles of around 180 degrees. You just can't get there. So proline is found in different secondary structural contexts than alanine. It's pretty comfortable in this weird area that we haven't seen a lot before. And there's a weird secondary structure there called a polyproline helix. I shouldn't say weird because it's actually really common, but collagen is has a lot of proline residues in it, and collagen tends to form what are called polyproline helices, which we won't talk about in any detail. But the point is the structure of the side chain, the fact that it's in a ring, makes predictable changes to the dihedral angles that the amino acid can adopt. So with that, I've said you need to begin memorizing the 20 amino acids, their one-letter codes, their names, their structures, and their side chain pKa's as appropriate. You'll notice there's a table in your text that gives you pKa's for all of the 20 amino acids. Most of that information is not going to be useful in the sense that they're going to tell you, for example, for this amino acid, which is called aspartic acid, they're going to tell you what the pKa for the carboxylic acid group on the backbone is. In general, that's going to be around 2. Each of the amino acids is a little bit different. The variation in backbone carboxylate pKa is not significant. So I don't care that you know that it's 2 point whatever for alanine and 2 point something different for glycine. 2 is close enough. Similarly, the ammonium group has a pKa of 9. Again, that's going to vary for each of the 20 amino acids. You don't need to memorize it. It's 9 point whatever for glycine and 9 point something different for proline. 9 is close enough. And in general, we aren't going to care that much about the pKa's of the backbone groups because most amino acids in a protein, as I demonstrated last time, are in this internal environment where they are bonded to an amide on the right and an amide on the left. Only the C-terminal and N-terminal amino acids are going to have free backbone atoms. So for the most part, I want you to focus in on side chain pKa's. We'll go through each of the 20 amino acids here, and you'll have to come up with whatever works for you to memorize things. I've grouped them in general categories, but these aren't hard and fast categories. They just are maybe to help you put them into groups so that you can help remember them. So alanine, as you know, is just the methyl group. Its one-letter code is A, because that makes sense. Valine is the one with the beta branching. The isopropyl group, side chain sort of looks like a V, so maybe that's a decent mnemonic device. The one-letter code is V for valine. Leucine is next. Leucine's a little bit different. Leucine has a regular non-branched beta carbon. Then on the delta carbon, we have branching with 2 methyl groups. So leucine is like valine if you were to insert another CH2 here between the alpha and the beta carbons of valine. Isoleucine is like leucine. Its one-letter code is I, only you take one of the methyl groups and you move it back over to the beta carbon, so there's branching on the beta carbon. One of the tricky things about leucine is this beta carbon is now a stereocenter, and probably most people forget what the configuration of that stereocenter is, and I'm not going to test you on the configuration of the beta carbon stereocenter of isoleucine. It is actually S, and that's the only one. There are diastereomers of leucine that have different, isoleucine rather, that have different configurations, at a different configuration at the beta carbon. We won't worry about that. But for completeness, I'm showing you the correct stereochemistry here. If you forget about it, it's okay. Methionine is the next amino acid, one-letter code M. Methionine is weird. It's grouped with the nonpolar amino acids. All of these in green are relatively nonpolar amino acids. It's got sulfur in the chain. It's a linear chain that goes out beta, gamma, delta, epsilon. So 1, 2, 3, 4 atoms after the alpha carbon. But the delta atom is a sulfur. Sulfur is not particularly electronegative, so this counts as somewhat nonpolar. This functional group's called a thioether. You don't necessarily need to know this, but if you ever got into protein biochemistry, methionine sulfur is easily oxidizable in air. It'll pick up an oxygen to form this, what's called a methionine sulfoxide. We deal with that in my lab all the time, so I'm telling you about it even though it's not that significant for you. But you'll need to recognize these as nonpolar amino acids. Okay. Next, we have some that are nonpolar but are aromatic. So this is phenylalanine. We're going to use the letter P for something else, but F makes a PH sound, so it's sensible for the one-letter code to be, or PH makes an F sound the other way around. But anyway, it's sensible for the one-letter code to be F for phenylalanine. It's got a beta carbon CH2 group and then a benzene ring. One of the things you need to know about benzene rings is that even though they are nonpolar, they're not, electron density is not evenly distributed over the whole ring. That is, sp2 carbons are more electronegative than protons, so the sp2 carbons of the benzene ring are electron rich and the hydrogens are electron poor. That means, and all of the, I'm showing you in pink individual bond dipoles associated with each of these CH bonds, where the plus end is over here and the minus end is over here. Those bond dipoles all cancel each other out, so there's no net dipole to a benzene ring, but the face is electron rich and the edge is electron poor. We call that a quadrupole. It's a distribution of electron density that's not a dipole, but it's still not uniform. And that's going to play a role in some things that benzene rings can do that other nonpolar side chains can't. Tyrosine, which we're going to use the letter T for something else, so we'll use the letter Y for tyrosine because there's a Y in tyrosine. This is BYU's favorite amino acid. Yeah, thanks. Not really. That was poor even by my standards. That was a 1 of 10 dad joke. Tyrosine is like phenylalanine, but with the para-OH group. So the side chain of tyrosine is a phenol. This is our first that has a side chain pKa that we need to worry about. Tyrosine has a pKa of 10. This OH group can be involved in hydrogen bonding, and sometimes you'll learn in biochemistry that this OH group acts as a nucleophile and gets modified with a phosphate group. The presence of the OH group is not really enough to make tyrosine be considered super polar. It's sort of intermediate between polar and nonpolar. I group it with these aromatic amino acids. Next is tryptophan, and here the rails sort of go off, or we go off the rails because we're now choosing a one-letter code that has nothing to do with any of the letters in the name. If you pronounce tryptophan as though it was 1930 and you were in a Looney Tunes cartoon hunting Bugs Bunny, like Elmer Fudd, you might say twiptophan, and so that might help you remember that the W, I don't know. In any case, the ring of tryptophan is more difficult to draw, so you will need to practice this. It's got a benzene ring and a 5-membered ring. This functional group overall is called an indole ring. It's aromatic. You can tell 2, 4, 6, 8, 10. The lone pair counts. Why? Because it's adjacent to a pi bond, so it's in a p orbital and part of the conjugated pi system. 10 electrons, that's aromatic. Like tyrosine, the tryptophan indole has this an NH bond instead of an OH bond in tyrosine. That can be a hydrogen bond donor. And like tyrosine and phenylalanine, the face of the tryptophan ring is electron rich, the edge is electron poor. And I'll have more to say about that in a little bit. Okay, any questions so far? Some of you may want to know what is the pKa of the indole NH. It's high enough that we don't worry about it at biological pH. All right, other questions? So histidine, now we're back to making sense, one letter code H. You've seen histidine before. We showed you a PLP-mediated biological decarboxylation to convert histidine into histamine, which is involved in allergic response. The histidine side chain is aromatic, so it could be grouped with the other aromatic amino acids, but unlike those other aromatic amino acids, histidine side chain can pick up a proton, and so at certain pHs, histidine can be positively charged. So I've grouped it with the other positively charged amino acids. It doesn't fit neatly in that positively charged group, but because it's more polar than the aromatic amino acids, it doesn't fit neatly in that group either. You can put it part of whatever group you want. You just need to remember its structure. So histidine has two forms that are relevant at physiological pH. You've seen this side chain before on your first exam. In this form, it's called imidazolium. It's aromatic. You've got lone pairs on this nitrogen that are participating in the pi system. So 2, 4, and 6. This is the nitrogen that picks up the proton. Actually, this imidazolium is resonance stabilized, so the positive charge can be delocalized on either nitrogen. And the pKa of this proton is 6. So at pH 7, the major form should be this neutral form called the imidazole form. And you can see why it doesn't fit neatly into plus or minus, plus positively charged versus neutral, because at physiological pH, it's about 90% neutral, 10% positively charged. So histidine is often found in enzyme active sites. It can act as an acid to lose its proton, or it can act as a base where this nitrogen picks up a proton. We'll see that a little bit later on when we talk about the catalytic triad in some proteases. Questions about histidine? Yeah?
Do you have those percentages swapped, or do I have it wrong?
At pH 7, if pH is higher than pKa, the proton should be gone. So if the pKa of this proton is 6, at physiological pH, that's a 1 pKa unit difference. That means equilibrium constant is about 10. So that's where we get the sort of 9-to-1 ratio. Or 10-to-1 ratio, whatever. Okay, others? Okay, so there are two other side chains that are always positively charged at physiological pH. One is lysine, and the other is called arginine. Lysine, again, we just use a one-letter code that doesn't make any sense, but we're running out of letters, and none of the other amino acids start with K, so whatever. I've gotten so used to it because I learned this, 25 years ago, that for me, K obviously means lysine. It's just in my head. I realize it's not that way for you, so you're going to have to work until you can have K associated with lysine. The other problem with lysine is it's tough to remember how many carbons are in the side chain. This is best done by practice. I remember just with my Greek letters that the nitrogen in the lysine side chain is on the epsilon carbon. So I count from the backbone alpha, beta, gamma, delta, epsilon. And then the thing that's bonded to the epsilon carbon is the nitrogen. The ammonium ion has a pKa of 11. So at physiological pH, this is going to be positively charged, and there would be perhaps less than 1% of the nucleophilic amine present. Even though it's mostly positively charged, you can see lysine act as a nucleophile in biology, and but for our purposes, if you think of it as always positively charged, that's fine. If it were to act as a base, you would need to first remove the proton with some other base, and then you could use the amine to do something. So that's lysine. Arginine is the other. You've seen arginine before on exam 1. Arginine is not an ammonium side chain. It's got a weird functional group that you may not know a lot about, and that's called a guanidinium group. So that's shown here. The first nitrogen of the guanidinium group is on the delta carbon. So arginine, instead of going out to the epsilon carbon, you draw alpha, beta, gamma, delta, and then the nitrogen's on the delta carbon. That nitrogen is attached to another carbon and then two other nitrogens, and this functional group is called guanidinium. It's positively charged, but you should be able to see that with the lone pairs on those adjacent nitrogens, that positive charge is delocalized by resonance onto all of those nitrogens. So some of you will look at the arginine side chain and say there's 3 different nitrogens that can be deprotonated. The answer is no, actually there's not. There's one positive charge that's delocalized. You can remove one and only one proton. You don't necessarily need to worry about which proton gets removed. This is the neutral form of arginine. In its neutral form, it's called guanidine. The pKa of the guanidinium ion is 12, so arginine is always positively charged at physiological pH. And its one-letter code is R. That's, again, we already used A, so now we use R, and it sounds like arginine, and now we get to use my other favorite amino acid joke, which is, what is a pirate's favorite amino acid? Arginine. Yeah, okay, yep, grown, that's the appropriate response, good, mission accomplished. So those are the positively charged ones. Remember, at physiological pH, lysine and arginine are positively charged. For our purposes, if you see histidine and pH is 7, you're going to consider it neutral because it's majority neutral. But you're going to be not surprised in biology to see the positively charged imidazolium do stuff because you'll recognize that there's about 10% of that at any given point. Okay, aspartate and glutamate. These are close cousins. And at this point, we're abandoning all correlation between letters and the names. So aspartate is D, glutamate is E. I don't know how you're going to memorize that. I'm calling them by their conjugate base forms because that's the form they exist in at physiological pH. In their protonated form, it would be called aspartic acid. And the pKa of the aspartic acid side chain is 4. Same thing with the glutamic acid side chain. The pKa of glutamic acid is also 4. So at physiological pH or pH greater than 4, you expect the majority to be negatively charged. For aspartate, the carboxylic acid is attached directly to the beta carbon, so carboxylate is gamma. For glutamic acid, the carboxylic acid group is attached to the gamma carbon. So the glutamic acid has one more CH2 in it than glutamic acid. D and E, not sure how you're going to remember that. One interesting thing about aspartic acid versus glutamic acid is it picks up some dihedral angles on the positive side of the Ramachandran plot for phi, whereas glutamic acid doesn't. It's able to adopt dihedral angles that a lot of amino acids don't. It's thought that that's because of the carboxylate group being close to the backbone. There's perhaps some hydrogen bonding with the backbone that can make some of that those dihedral angles more favorable. Let's see. So those are the charged ones. You've got positive and negative. Again, though I've included histidine here, if you see it in an amino acid sequence and the pH is 7, you're going to consider histidine to be neutral, but you'll consider lysine and arginine to be positively charged, aspartate and glutamate to be negatively charged at pH 7. All right, next we've got polar but neutral. This is serine. You've seen serine before. It's got an OH group. The pKa of an alcohol is around 16. That's maybe not going to be in the table that's in your textbook. It might, I'm not sure. But I want you to know it. Of course, you probably already know that because you know pKas of alcohols. We will see serine sometimes get deprotonated and then act as a nucleophile. So that's why I want you to know that pH. One-letter code makes sense, so that's not too much trouble. Threonine you've already been introduced to. It's got beta branching, which makes it more favorable in beta sheets. Likewise, the pKa of threonine is around 16. And you'll sometimes see threonine act as a nucleophile. Both threonine and serines can get modified with phosphate groups. You'll learn more about that in biochemistry. Another one that is strange is called cysteine. Cysteine is the sulfur version of threonine. So instead of an OH group, you've got an SH group. The presence of the sulfur lowers the pKa because sulfur is larger than oxygen, and so in the conjugate base, the negative charge is spread out more. And so cysteine can easily exist in its negatively charged thiolate form. Cysteine is the most nucleophilic amino acid, but it's the fact that it's sulfur has something else that can go on with cysteine, and that is the cysteine side chain is oxidizable. You can make disulfides with cysteine side chains. So in a disulfide bond, one sulfur from a cysteine is connected to another sulfur. The hydrogen is now gone. We won't necessarily worry about showing you how that oxidation reaction happens. It can happen spontaneously in air. And this is important for structural stability in all kinds of secreted proteins antibodies, cell surface receptors, insulin, other peptide hormones often have disulfide bonds that provide structural stability. This isn't such a big thing nowadays. It maybe is coming back. I don't know. It was definitely a big thing in the 80s. People would go, and if you had straight hair, you'd go turn it curly by getting a perm. You guys have heard of that? No? Vaguely, sure. Well, one of the things they're doing to make that happen is they're reducing a bunch of disulfide bonds in your hair, in the keratin in your hair. Then they're twisting the hair around, then they're reoxidizing, and those disulfide bonds tend to hold the new shape of the hair in place. And that's why some of the chemicals used to do that, you'll smell a little bit of sulfur, and they're using thiols to do the reduction chemistry. Okay, 2 more in the list of polar but neutral. These are related to aspartic acid and glutamic acid. They have the same shape, only the OH of aspartic acid is replaced with an NH2 for asparagine. That's why the names are similar, aspartic acid or asparagine. Asparagine has an amide instead of a carboxylic acid. The 1-letter code is N. Asparagine, like aspartic acid, opens itself up to more dihedral angle options in the right-hand side of the Ramachandran plot. And that's again thought because you've got these hydrogen bond donor and acceptor groups that are pretty close to the peptide backbone, and so they can stabilize conformations that most other amino acids can't. Glutamine is like asparagine, only it has 1 extra CH2 unit. The 1-letter codes here, again, don't make any sense, N and Q, so you're just going to have to remember them. I suppose asparagine has an N in it, so there you go. Lastly, in this last category, I have the 2 weird ones, and they're weird for reasons we've already discussed. Glycine, 1-letter code G, doesn't have a side chain, so it's way more flexible than the other amino acids, can totally populate areas of the Ramachandran plot on positive values for phi. Glycine, because it's so flexible, often doesn't work well in helices. It introduces too much flexibility in the backbone, and so glycine will often break helices. Proline as well is weird because it's got this cyclic side chain that we've already talked about and restricted conformational freedom. Proline is, even though it's got dihedral angles that would be comfortable in an alpha helix, proline doesn't work well in an alpha helix because alpha helices involve hydrogen bonds, and proline's nitrogen is incapable of being a hydrogen bond donor. It lacks the NH group. So proline is also not found a lot in helices. Both of these are often found in places where secondary structure is about to change. So if one helix needs to stop and turn around and form another helix, proline and glycine are often found in those turns. If one beta strand needs to stop, turn around, and turn into another beta strand, glycine and proline are often found in those turns. Okay, so that's your survey of the 20 amino acids. Anything else you want to know? So one question that usually comes up at this point, somebody doesn't want to ask it but certainly has it, is all those things you told us about the amino acids, are we supposed to memorize and regurgitate them? As you probably observed, this class is more about what you can do than it is about recall of facts. So given data, I expect you to be able to use the data to draw conclusions. But in terms of memorizing, you absolutely do need to know names, 1-letter codes, side chain structures, and side chain pKa's. Okay? All right. Well, now I want to talk about the non-covalent interactions that are available to peptide and protein side chains. Things that you can see happening, things that help proteins adopt their particular secondary and tertiary structural shapes. So this is going to be a review of things you learned in 351, but it's probably been a while. And I do want you to be able to think through some of these things sensibly. Understanding these non-covalent interactions is another key towards understanding the relationship between sequence and structure and function. So understand that non-covalent interactions are considered weak in the sense that they're not as strong as covalent bonds. They're worth about 1 to 10 kilocalories per mole, meaning it takes 1 to 10 kilocalories per mole of energy to break one of these non-covalent interactions. That's about 10 to 100 times weaker than, say, a carbon-hydrogen bond. So they're weak, but if you remember the number I told you, I think, before, that a difference of about 1.4 kilocalories per mole is worth a factor of 10 in equilibrium constant, you can see how non-covalent interactions could dramatically influence equilibrium constants for a process. Like, for example, protein folding. So here are some examples of non-covalent interactions that happen in peptides of proteins. One is called a salt bridge. That's a fancy term for an ionic interaction. So if you have a lysine side chain, which is K, this is the alpha carbon, so beta, gamma, delta, epsilon, and there's the side chain. There's the ammonium group. That positively charged ammonium group can interact with a negative charge on, say, a glutamic acid side chain, which would look like this, where the carboxylate is is attached to the gamma carbon. This is glutamic acid, 1-letter code E. So E and K can interact in a favorable way. Other amino acids that could do this on the positive side would be R, and other amino acids on the negative side that could do this would be D. So when you see Ks and Es, you can be thinking, those are oppositely charged. It's possible they could interact with each other in a favorable way. Usually the strength of that interaction is dependent on how close the amino acids are to each other. The closer the charges are, the stronger the interaction. It's basically a Coulomb's law type relationship. Another type of interaction that you should be familiar with is hydrogen bonding. This can occur, there's tons of examples of this all over the place. This can occur anytime you have an electronegative atom near an atom that has a partial positive charge on it. So we can't here enumerate all possible examples of hydrogen bonding in peptides and proteins, but you're basically looking for electronegative heteroatoms and bonds between hydrogen and an electronegative heteroatom. Here's an example. This type of thing occurs in an alpha helix. So here is one amino acid and another amino acid. And then this nitrogen on one amino acid can be hydrogen, the NH bond on one amino acid can be hydrogen bound to an oxygen on another amino acid. Whoa, that eraser was too big. So the dotted line here represents a hydrogen bond. Basically what you've got is a dipole between nitrogen and hydrogen that puts a positive partial charge on the hydrogen, another dipole between carbon and oxygen that puts a partial negative charge on the oxygen, and the partial negative charge on the oxygen is attracted to the partial positive charge on the hydrogen. Some of these can be quite strong and even partially covalent, but that's sort of what a hydrogen bond looks like. That should mostly look familiar to you. Any questions about that? Okay, this might be new, and this is kind of cool. I'm going to call these pi interactions. And I don't think these are discussed in your text. Hydrogen bonding and salt bridges certainly are. Pi interactions are cool, you can understand them, and I just can't resist. So since it's my class, we're going to do it. All right, so imagine, I'm just going to illustrate pi interactions with benzene rings because that's going to be easier. We're not going to worry about where the phenylalanine or tryptophan or tyrosine side chains are. What I say about benzene rings also applies to the aromatic side chains of tyrosine Y, tryptophan W, and phenylalanine F. Okay? So as I pointed out previously, if you look at a benzene ring sort of on its edge, the electron, the sp2 carbons are more electronegative than hydrogens. We're going to use the color blue for, let's use the color red for electron poor. The edge of the ring is electron poor, and then the face of the ring is electron rich. And that means that you can no longer think of benzene as being totally nonpolar. It doesn't have a dipole, but you have to think of it as having an electron-rich face. So one of the things that can happen is a cation, say a potassium ion, or perhaps a positively charged amino acid side chain. Maybe I'll illustrate that with a positively charged amino acid side chain. One of the things you can see in proteins is the positively charged side chain from a lysine residue sitting down close to, within contact distance of the electron-rich face of a phenylalanine residue, or a tryptophan amino acid, or a tyrosine amino acid. And what's going on here is, as I said, the face of the benzene ring is electron rich. The cation is electron poor, it's positively charged. So you have basically a charge interaction between the full positive charge on the nitrogen and the electron rich surface of the benzene ring. We call that a cation pi interaction, and those are found all over in proteins. They're actually important in acetylcholine receptors and acetylcholine esterases, and they're actually found tons and tons in proteins. Most proteins have at least one cation pi interaction. Another thing that can happen is that the electron-rich face of one benzene ring, say on a phenylalanine residue, can interact with the electron-poor edge of another benzene ring. So we call this sort of T-shaped stacking, where the electron-poor face of one aromatic ring, the electron-poor edge, rather, of one aromatic ring gets close to the electron-rich face of the other aromatic ring. And again, opposite charges attract, even if they're partial charges. So we call this an example of sort of T-shaped pi stacking. Another thing that can happen is offset stacking of two aromatic residues. So let me see if I can illustrate this. Here is one benzene ring. It's a phenylalanine side chain. And I'm going to draw the hydrogens here. Again, illustrating the electron-rich face and the electron-poor edge. It would be probably, and then I'm going to copy this. You can imagine that if I stacked two benzene rings close to each other, one directly over the other, that should be repulsive, right? Because the positive edges would repel each other, the negative faces would repel each other. So instead, what tends to happen is an offset stacked example where they'll hover together like this, such that the electron-rich face of one is over the electron-poor edge of the other. This is called offset pi stacking. And so you see these things all the time in proteins: hydrogen bonding, salt bridges, pi interactions. We haven't even illustrated all possible hydrogen bonding. You really couldn't. Anything that's capable of hydrogen bonding you see in proteins. All right. Any questions about that? So we need to do one more thing, and this is perhaps the most important of all of the non-covalent interactions in peptides and proteins. It's almost maybe not an interaction in and of itself, and that's something called the hydrophobic effect. The hydrophobic effect is not the same. You've learned about this before. The hydrophobic effect is not the same as induced dipole or van der Waals interactions. So van der Waals interactions are induced dipoles. They occur with everything, but we think of them as most significant when you're dealing with side chains that don't have a lot of polar groups in them. And you learned in 351 that non-polar groups can interact with each other via these dispersion or van der Waals interactions where one molecule distorts its electron density temporarily and the other one does too, so that their positive and negative sides are complementary to each other. And you've learned about that. Those interactions are real, they are relatively weak, they can add up to a lot. People tend to conflate in their minds van der Waals interactions with the hydrophobic effect. They use, those strong van der Waals interactions are why oil doesn't dissolve in water. Not exactly, because the van der Waals interactions are weak. You may have learned about this in 351. If you didn't, it may be a revelation to you today, but it's important for you to understand because it's at the basis of almost all conformation issues in peptides and proteins. So the idea is I'm going to draw two images here, two cartoons. Here is an unfolded protein, and then here is a folded protein, all right? That's terrible, I know, but it's a cartoon. It's not even a good cartoon. We're going to draw some nonpolar side chains at various positions. I'm just drawing a few of them. And then I'm going to draw some polar side chains with some positive and negative charges at various places, okay? What happens when nonpolar, and I'll highlight in gray the nonpolar functional groups. What happens when nonpolar functional groups are exposed to water is that they don't interact well with water, and so the water molecules in the immediate vicinity of the nonpolar side chains, suddenly they have space around them that's not filled with water molecules, nothing to interact with, so they correspondingly interact with each other much more favorably. And the hydrogen bonds in the immediate vicinity of the nonpolar side chain get stronger, and the water molecules start to slow down in the immediate vicinity of the nonpolar side chain. In fact, being in the immediate vicinity of the nonpolar side chain causes the water molecules to lose entropy. Sorry, I was going to say loose entropy. Lose entropy. And in fact, around each nonpolar group, you have a similar sort of ice-like cage of water molecules. And the reason they form this ice-like cage around the nonpolar stuff is, again, they can't interact strongly with the nonpolar stuff, so they interact correspondingly more strongly with the water molecules that are immediately around them. That's what happens when nonpolar surface area is exposed to water. So this is what happens for a protein that is unfolded. In contrast, in the folded shape, usually all the nonpolar side chains are buried in the interior where they do interact with each other via van der Waals interactions. So you do pick up van der Waals interactions in the folded protein. That's probably not the major effect though. The major effect is all of these water molecules that used to be in the immediate vicinity of the nonpolar side chains are now released, and they have much increased entropy, which is why folding is downhill. Folding has a negative free energy, but in a lot of cases it's driven by entropy, because when the molecule's unfolded, the water that surrounds the protein loses its entropy, loses quite a bit of entropy. When it folds, those water molecules are released and they're saying, we're free. And that is the origin of the hydrophobic effect. So when you look at protein structures, you will see that putting nonpolar stuff in the interior away from water is the major driving force for protein folding. And it's an important consideration we're going to return to again and again as we try to tease out the relationship between sequence, structure, and function. So you got some general principles that are emerging so far. You want complementary charge interactions between side chains or backbone groups. You want salt bridges, you want cation pi interactions, you want hydrogen bonding, and perhaps even more importantly, you want nonpolar stuff in the interior of the protein, not because the van der Waals interactions between the nonpolar things are so strong, but rather because by putting the nonpolar stuff in the middle of the protein, you release water molecules and increase their entropy, and that makes folding favorable. All right, so in the time we've got left, I'm going to write a sequence of a protein, and we're going to start trying to understand how that sequence relates to the structure of the protein. Sorry. Okay. So this is a real peptide sequence or peptide or protein sequence that I've made before. For this particular sequence, it's been modified such that the N-terminal group is tied up in an amide bond. This is an acetyl group. That doesn't happen a lot in nature, but this is a synthetic peptide that we've made in my lab that other people have made. So this nitrogen is the N-terminal nitrogen. One thing you need to know about peptide sequences, they are always, always, always written left to right, N terminus to C terminus. Always. You need to understand and know that always, because at some point I'm going to try to trick you on it. So always. So, all right, this nitrogen here belongs to the N-terminal amino acid. So that nitrogen belongs to the R. RMKQ, LED, KVEEL, SKNY, HLENE, VARL, KKL, VGER, and then the C-terminal R has a free carboxylic acid, which at physiological pH should be in its negatively charged form. Okay, what you need to do is I want you to go through that sequence and I want you to identify every amino acid that should have a positively charged side chain, every amino acid that should have a negatively charged side chain, and then I want you to identify every amino acid that's nonpolar. Okay? Those three things, and you're going to see next time how those three things lead to the interesting structure that this peptide adopts. All right, with that, we are done. Have a good Wednesday. I'll see you Friday.